Hello guys, let's start with another YouTube lecture for you. This is again a very important topic that is called as epigenetics that suggests the effect of the environment on the gene expression. How exactly the gene expression will change in the presence of different environmental conditions. For example, if you take the two identical twins, they have the same genetic material, but the way they develop, the way they have developed the intelligence level or their normal behavior, that is going to be different from each other. Or maybe the some other cases, if you change the climatic condition or you move from one place to another, certain changes occurs in your body. Some of the changes are really very stable that might go to the one generation to the other. That could be the heritable changes, but some changes they may not go from one generation to the other. It all depends whether these changes happened on the somatic cell or the germline cell. If it is on the germline cells, it might carry from one generation to the other. And if not, it will not go from one generation to the other. Let's talk about exactly what is the epigenetics is all about. I'll suggest the few things I'll show you. So what is epigenetics? So this is, if I go by the definition, so this is the changes which is happening in the phenotype, not in the genotype. Okay, without changing the structure of the genome. And this, the structure, these changes, as I told you before, it might be inherited or might not be. So how exactly these changes are going to happen? So what exactly the epigenetics says? It represents the science for the studying of the heritable changes of the DNA, not involving changes in the DNA sequence. The DNA sequence is not going to be changed. Rather, it is going to happen on those regulatory element which might change the gene expression. Okay, hopefully these things are very clear to you. Let's talk about the genetic information and the epigenetic information. We'll try to make it different, and we'll do some kind of comparison as well. First, let's talk about the genetic information. It is the one which provides the building block for the manufacture of all kinds of proteins needed for the functioning of the cell. Whereas epigenetic changes, they provide the additional instructions on how, when, and where these information should be used. Now, there are the few things which is very, very important. There are the two changes which is happening for epigenetic information. One is going to be on the DNA. Usually the DNA has just one chain that is called as methylation and the demethylation, whereas the stone modification, which is post-translational in nature, which is of 5 different types: acetylation, methylation, ubiquination, summolation, and phosphorylation. And these changes are going to happen on the long N-terminus tail of the histone. This is the basic idea about the nucleosome model. If you look at the how exactly our chromosomes are organized, so you can start from the DNA, which is around 2 nanometer in diameter. It is going to wrap around octamer of the histone proteins S2A, S2B, S3, and H4, and H1 is going to play a role of the linking them and condensing them. Then they're going to be organized in the form of zigzag structure, will make the 30 nanometer structure, and then they'll be converted to the loop domain, which requires non-histone proteins as a scaffold protein at the bottom, and then they'll be organized into the 1400 nanometer, a proper chromosome, and it's one of the arms. Now let's see where the changes are going to happen. Before I go into that, if you separate out the nucleosome or the histone, which is octameric structure, and around 146 base pair of the nucleotide is going to be wrapped around, and it has the 4 types here that is shown, and one which is not the part of the nucleosome, rather it is the on the linker DNA, which is not shown in this. And these the tails I was talking about, the long N-terminus tail of the S3, H4, even the S2 and S2B, but largely the changes are going to happen on the H3 and H4. And this is how it is going to happen. Look at over here. Here the H1, which plays a very important role in condensing these beads on string structure into the 30 nanometer fiber. And these are the changes which are happening, there are many different kind of proteins which play important role in condensing them in a further more condensed form. And this scaffold I was talking about in the loop domain, which will turn into the 300 nanometer structure, and many other enzymes which are going to be there. For example, the stone modifying enzymes, chromatin remodeling complexes, RNA polymerase, all they're going to bind on this. This is the methylation, which is very, very common, especially on the cytosine residue, because unlike the prokaryotic system, in the eukaryotic system, we have the DCM methylase, which is going to methylate the cytosine at the 5 position. So 5-methyl cytosine is going to be present. And if you look at these structures, which are very common on the CpG island, these are the dinucleotide structure, maybe present in the hundreds of number, some 100 to 150 bases upstream from the transcription start site. And if they get methylated, they will interfere, to bind with the transcription factor and the RNA polymerase as well. So whenever the methylation occur in this region, those regions are not going to express. That means genes are going to be silenced. And these methylation can occur de novo as well, or it can be used, as it has in the main genome sequence. That means to maintain it. Now there'll be 2 ways: the de novo, these are the new changes, and maintenance, which is already they have, in the template. And sometimes the demethylation occur because of some of the natural factors or the environmental factor. And many other enzymes, for example, DNA methyltransferase, we have the methyl CpG binding protein. They are one which is going to lead to and at the same time, the stone deacetylase, which removes the acetyl group from the stone proteins, that could lead to the chromatin condensation and gene repression as well. There's one more protein which plays, important role in the condensation, is the condensin. So some cases, in the case of C. elegans, it has been seen. So chromosome specific condensin, they can condense the chromosome and eventually lead to get converted into the heterochromatin, and gene will not express. These are the certain changes we call the stone code hypothesis, or the stone code, which could be written, which could be read, and which could be erased if it is not required. So for example, it is related with switching of the gene and switching off a gene. If the acetylation phosphorylation occur, gene is going to be switched on, whereas the methylation eventually lead to the switching of a gene or gene repression. These are the certain proteins which are involved. Name wise, acetylation is done by the histone acetyltransferases, HATs. We have the different example. One which is very common is the GCN5. Similarly, the deacetylases, which is class 1 and class 2, methylation, both, which is having the lysine set domain and HMTase, and non-set domain HMTase, that is also called a DOT1, and arginine PRMT family or CARM1. Demethylation is usually done by the LSD1 protein, and ubiquitinylation, whenever a protein is going to be ubiquitinylate, that means the protein is going to be dead, or is going to be removed, or going to be denatured. And deubiquitination is going to be, a protein because it's saga associated with the UBP tag. Now, what exactly this hypothesis means? Just now I told you, it will just let us the gene know whether it is going to be expressing themselves or not. So the acetylation will take the gene to switch on, whereas the deacetylation and the methylation will lead to the silencing of the gene. These are the 2 basic differences between the heterochromatin and the euchromatin region. Heterochromatin is one which is not transcriptionally active, and that is condensed region, whereas euchromatin region is the one which is loosely attached and organized normally chromosome into the 30 nanometer fiber during the interphase, and very, very active transcriptionally. And this region, whether it's a heterochromatin or the euchromatin, they're going to be synthesized in the interphase that is called as S phase. If you see this HP1, which will always look to the region which is methylated and going to make this region very, very inaccessible to the RNA polymerase and other regulatory regions, which is found in the heterochromatin, whereas the euchromatin has a lot of acetylated region. Now, one change I'll show you, which is the probably the best example of the X chromosome inactivation. Again, this happens during the embryonic process. Out of the two X chromosome in the female, one they get it from the parent, which is parental in nature, and there is a maternal in nature. But one which is going to be inactivated, that is not very sure. But out of the two, one is going to be inactivated. Basically, because of the region we call as there's one RNA created XIST, which is going to bind to the 3' terminus to make it inactivated. At the same time, some of the other factors which is playing a very important role in the condensation of the chromosome. I told you in condensation we have the chromosome specific condensations that are going to play a very important role. Now, another important thing I need to tell you is that why this region, why this epigenetics is very, very important. That the certain changes which we need, which we can induce, in a plant or in an animal, we can bring it about. Especially the plant, there are a lot of research are going on where they are, the bringing about the certain changes epigenetically. So study of the epigenetics is sometimes very, very important to understand the molecular biology of the organism and many changes which is happening, like the cancer or maybe the aging process, maybe other factor, or maybe development of the certain cancers or the some other diseases which is linked to the epigenetics. Hopefully this lecture had helped you and you enjoyed it throughout. And for any curiosity or any queries, you can write your comments, or you can ask me the question directly in the comment box. Thank you. Thank you for listening me patiently. Thanks a lot.